All right, guys, well, welcome to week 6. This week we are in our last week of dealing with biology, and we are studying the eye, the ear, a little bit of our olfaction, and a little bit of our nose. So after we get through this week, we are done with biology, and we start our theory-based, which is really exciting. But sensation and perception is always an interesting week, and it kind of changes the way you think about everything. So here we go. So what is sensation? Now, sensation is the activation of receptors of various senses organs. That is your nose, your ears, your eyes, your mouth, and your kinesthetic or your skin. Now, your sensory receptors are a specialized form of neurons, as we've been talking about. They're the ones who send your afferent neuron to the brain. As you can see, all of AP continues to build on each other. Now, our sense organs are eyes, ears, nose, skin, and taste buds. Now, when we talk about sensations, there are different amounts that our eyes and our ears and our taste buds can all pick up. Now, when we start talking about sensory organs, we have to talk about sensory thresholds. Now, when we talk about sensory thresholds, we have to talk about just noticeable differences, or J&Ds, or difference threshold. All mean the same thing. And what it means is the smallest difference between two stimuli that is detectable 50% of the time. What that means is, if I ask you to pick up a fine 5-pound weight, and you pick it up, it's going to be fine. Okay, you're going to pick it up, you're going to lift it up, and put it back down. However, if I give you a 5.5-pound weight, are you really going to notice the difference? Probably not. However, if I have a dime and give you 2 dimes, you're going to notice the difference? Absolutely. What that means is there's only so much that our senses pick up in difference to one another. If it's a large enough difference, we can detect it. If it's a small difference, like for instance, a 1/2 pound to 5 pounds, is really not that much. However, twice as much weight to something that is so small and minimal is a big deal. So keep in mind, we have to be aware of what we can and can't tell the difference of. Okay? Now, sensory thresholds also deal with absolute thresholds. Now, absolute threshold is the smallest amount of energy needed for a person to consciously detect a stimuli 50% of the time. Now, just noticeable differences, or J&D, is by each person individually. Each person can only sense so much. Some of you have better sense of taste than other people. Some people have better eyesight than others. All of that depends on by person. Absolute threshold is by the best person who's ever done it. So, for instance, there's examples in the chart below. So, sight, there has been one person who has seen a candle 30 miles away on a clear, dark night. I cannot see a candle 30 miles away on a clear, dark night. I can't. But one person has. That's a sense, and that's an absolute threshold. Absolute best someone's ever done something. Okay? So absolute threshold is the best someone's ever seen, heard, smelled, tasted, touched. Just noticeable difference is how well you as an individual pick up information. Okay? So absolute threshold. I mean, those things are fascinating. Like, for instance, someone has been able to detect a bee's wing falling in a cheek from 1 centimeter above. Like, who notices that stuff? It's crazy. Anyway, so another major component of sensations is subliminal sensation. Now, subliminal stimulation is stimuli that are below the level of conscious awareness. Our body, our sensory receptors are picking up all the information that is going on around us. So it's picking up all the smells. It's picking up all of the sights, everything you're seeing. It's picking up all the things you're hearing, all the things that are touching your skin right now. But think about how overwhelming would that be if we had to process everything. So right now, I have Duncan trying to harass me while I'm making this video for you. And I'm sitting in the room with 3 candles going. I have a litter box in the other room. I made lunch a little bit ago, and I have some crackers next to me, and all of these scents are going on around me, and my brain cannot function with all that information. So what it has to do, it has to limit how much information is getting to my brain. Because frankly, if I had to process all the different smells, all the things going on around me, I wouldn't be able to make this video. So what our brain does is it tries to figure out how much of each to bring in. Now, if If it's just strong enough to activate a sensory receptor, but not strong enough for people to be consciously aware of them, there's a threshold. Subliminal means below threshold. So there are certain things right now that are below my threshold. Like, for instance, probably the smell of my litter box, because I just changed it today, so it's clean, so it's not that big of a deal. However, this morning when I cleaned it, it was above threshold. Now, subliminal sensation deals with the process by which subliminal stimuli act upon the unconscious mind. Now, I may not be able to I may not be consciously smelling all these things. However, all this information is being processed in my brain, and it does influence us. Like, for instance, when I'm sitting here and McCrae had crackers earlier and I can kind of still smell the crackers, I am now more hungry than I would be if I didn't smell the crackers. All of those types of things are going to influence, and that's called subliminal perception. When these sensations, these subliminal stimuli, trigger our perception or trigger our change in behavior. Now, there is absolutely no evidence of subliminal stimuli in advertisements that appear. So, for instance, we've all heard of movies, theaters showing pictures of popcorn and soda and all that stuff during the films. There's no evidence to say it works on everyone, but there is evidence to say it does work on some people. Typically, your weaker mind people typically fall for it. Now, habituation and sensory adaptation. Now, habituation is the tendency of the brain to stop attending to constant, unchanging information. Like I said, there's so much going around in my house, even though I'm the only one here with two dumb cats. What we're going to see is that there is a lot of things going on in my house right now. So, your brain can't process everything. So, consistent things, consistent smells, consistent hearing things, we kind of tone out. For instance, if you walk in the house and your trash barrel is full and stinks and reeks, when you walk in the house, it's like it gets smacks you in the face with the bad smell. However, after being in the house for 15, 20, 30 minutes, you don't even smell it. That's your senses. Once you walk in the room, they say, oh my God, it smells, it smells, it smells, it smells. Then all of a sudden, it's saying, okay, well, clearly you don't care that it smells, so I'm tired of telling you. Now, sensory adaptation is the tendency of sensory receptor cells to be less responsive to a stimuli that is unchanging. Now, habituation is when your brain stops telling you. Sensory adaptation is when your sensations just stop telling you. Like, for instance, have you ever just been cold? And then at some point you just reach this whole level of just being cold. Like, you're just cold. You're not like, oh my God, you're shivering. You're just cold because your sensations are tired of telling you that you're cold over and over again. Now, microsadies are constant tiny movement of the eyes that prevent sensory adaptation to visual stimuli. The thing is that our eyes are what we depend most on sensations. So we depend heavily on this for our evolution, for all of other things we do. So we don't let our eyes ever have sensory adaptation because our eyes are constantly adjusting and shifting. That way, every once in a while you do zone out and you're kind of unaware, but it's not your eyes zoning out, it's your brain zoning out. Okay? Now, psychological aspects to light. There's different types. There's brightness and color. Now, brightness is determined by the amplitude, or how fast the light is moving. Now, how high or how low the wave is, the higher the wave, the brighter the light, the lower the waves, the dimmer. So depending on how fast the light waves are going depends on the brightness. If it's nice and slow, it's very dull. If it's very quick, it's very bright. Now, color or hue is determined by the length of the wave. Is it a short burst or is it a long burst? Now, long wavelengths are found in red and are at the red end of the visible spectrum. So the whole spectrum of light is visible to the human eyes, where shorter lengths are found in the blue end. So, blue is going to have your shorter waves, red is going to be your longer. Now, saturation refers to the purity of the colors people see by mixing gray or black would lessen the saturation. Okay, now you do need to know that amplitude gives you brightness, and you do need to know the hue is determined by wavelength. Please be familiar. Now, as you've seen with rainbows, hopefully, and your science teachers probably talked to you about the visual spectrum, how white light, you put it in a prism, and then all of a sudden it breaks it down into all the lights. Okay, all of these we can see. Now, in class, we've been working on the structure of the eye, and we talked about it in class and we've gone through. Now, the cornea is the clear membrane that covers the surface of the eye and protects it. It's what you touch when you put your contacts in. Now, the radial keratotomy is a vision improving technique. They use this fact by making small incisions in the cornea to focus the eye. You don't really need to know it, but do you know it's done through the cornea? Now, the aqueous humor is the next visual layer, clear, watery fluid that is continually replenished, and this is going to keep your cornea moist. Now, the pupil is a hole through which light in the visual image appears through the interior of the eye. It is actually not a structure, it's the lack of a structure, it's literally just a hole. Now, the next thing is the iris, which is a round muscle, the colored part of the eye in which the pupil is located. Remember, the pupil is made out of the iris's muscle that can change any size. It controls how much light is getting in and helps focus the image. Now, the next major structure that it goes through is the lens, which is a clear structure behind the iris, which is held in place by muscles, which is focusing. It focuses all the images coming through. Now, visual accommodation is done by the lens. It is the changing of the thickness of the lens as it focuses on the objects. So if you need to like squint to see something because it's so small, it's visual accommodation. If you need to like kind of open your eyes a little bit bigger to see everything, that would be also visual accommodation. So it's pretty important. Now, your vitreous humor is a jelly-like fluid that nourishes the eye and gives it its shape. Okay, it's going to be inside the eye. It's what we labeled in the center of the eye. Now, the most important step of your eye is retina. Now, it's the final stop for light in the eye. So it goes through the cornea, through the pupil, surrounded by the iris, passed through the lens into the back of the eye. Now, the retina has 3 layers of cells: ganglion, bipolar, and photoreceptors that actually respond to the light. And the photoreceptors are technically your retina. Now, on these photoreceptors, you have rods and then you have cones. Rods are processing all your gray and black light, while your cones, C for color, is processing your color vision. Now, another major part of your retina is going to be the blind spot. Now, it is the area in the retina where the axons and the 3 layers of retinal cells exit the eye to form part of the optic nerve. And it's insensitive to light, so it will not process any light. Now, interesting. All right, and here is your eye. As we did the structure in class as we labeled everything. Now, this is a very important picture. Remember, it goes ganglion, bipolar, then your rods and cones, which are your photoreceptors. Okay? Now, as we get through the eye, you need to understand that the left and right visual fields are sending both information to both sides of your brain. It goes through your optic nerve and then goes to your optic chiasm. Then the images are matched together from your left and to your right, and then it's put in your visual cortex. Now, one of the most challenging aspects of the eye is your theories of light. And we'll be discussing that a little bit more in class, specifically on Monday and Tuesday. So how does the eye work? Okay, now dark adaptation is the recovery of the eye's sensitivity to visual in darkness after exposure to lights. So night blindness would be an example. It's when your eyes adjust to the night and kind of you're still able to kind of see. Even though it's really dark, you're still able to see after a while. Now, light adaptation is a recovery of the eye sensitivity to visual stimuli in light after dark. So after you walk out into the sun, after like leaving the movies, for instance, and it's like super bright and you're like, oh my God, it's so bright. Okay, that's called light adaptation. Now, when we're dealing with color vision, we have different theories to process. Now, your different theories, the first one is your trichromatic theory, which is also known as the Young-Helmholtz. It's a theory of color color vision that proposes 3 types of cones: red, blue, and green. And these 3 types of cones are the only light colors we can see, and their blending creates the other ones. Now, using this, some of the information that we use to kind of prove this theory is after images. Now, after images is the image that occurs when a visual sensation persists for a brief time after the original stimulus is removed. What that means is that if you stare at something for quite some time, eventually it will change colors in front of you. Now, another major theory is the opponent process theory. It's a theory of color vision that proposes 4 primary colors with cones arranged in pairs. So red and green are together and yellow and blue are together. And all the light of those two are brought together and then those are mixed. Okay, so you have 2 major theories, the trichromatic 3 theories of red, blue, and green, and then your opponent process theory, which has green, red, and blue, and yellow and green, blue and yellow together for the first time. Okay. Now, if you look at after image, so if you stare at the white dot at the center of this oddly colored flag for about 30 seconds, now look at a white piece of paper or white wall. Notice that the colors are now normal, expected colors of the American flag. This is also primary colors. So if you stare at that white dot for about 30 seconds really intently and look away at a white wall, it'll show you the right colors, which is pretty cool because your brain's processing that information with its buddy. So the yellow matches up with the blue and the red matches up with the green. Now, color blindness, or is also known as microchrome blindness, either have no cones or have cones that are not working at all. Okay. Now, red and green blindness is probably your most popular. Either the red or the green cones are not working and picking up and receiving color. We do know that it's linked to sex linked. So men are actually like 4 times more likely to have color blindness than women, which is pretty cool. So here's an example. Now you have a person on the left. That's what a person would see if you have no color processing. Although on the painting on the left was done with a person with normal or trichromatic, okay? And then the one on the right so the one on the way left is monochromatic. So if someone saw the picture, this is what they would see if they had no cones. Person on the right is done with red or green colors. The one in the center is what we would see as normal viewers if you don't have any problems with color vision, which is pretty cool. Now here's another color test that we do. If you can see the numbers inside, to the left it's an 8, to the right it's a 96. If you have difficulty seeing those numbers inside those colors, that could tell you is one of the tests we use for color blindness. Now, sound is dealing with wavelengths, which is frequency, or pitch is high, medium, or low. Now, amplitude is interpreted as sound, how loud or soft the sound is. Purity is interpreted as timbre or the richness of a tone. So depending on how much vibration it really has. And a hertz is how we actually measure it, which is pretty cool. Now, when we deal with sound waves, we're dealing with different amounts. Now, when we deal with sound waves, for instance, normal breathing on our hertz scale is a 15. A quiet office is a 40, so a quiet classroom is a 40. A vacuum cleaner is a 70, and a jackhammer 3 feet away is 125. As you can see, there's different wavelengths that tell us how much pain would be created. Now, the structure of the ear, we have the auditory canal, which runs into your tympanic membrane. We have the eardrum, also known as tympanic membrane, which is covering and converts the sound waves into physical movement by going through your ocelles, hammer, anvil, stirrup. We're also going to have the structure of the ear, which is cochlea and organ of Corti, which rests in the basilar membrane, which contains your receptor cells for hearing. Now, in your cochlea, you're going to have your basilar membrane. Your basilar membrane is going to be processing all of your sounds, and it's converting the movement of your cilia in the mucus of your cochlea into neural impulses. Now, the neural impulses will escape your cochlea into your auditory nerve, which is just a bundle of axons covered, and we'll take it from there. Now, we did our example in class of all your different ears and all those for you. Now, your pitch is you're going to be higher sound waves. So the higher it goes. Now, place theory is a huge part of your hearing theory, and it is based that different pitches are experienced by different hair cells along the organ of Corti or along the basilar membrane. So your lower pitches are going to be held in the lower, while your higher pitches are going to be held in like the more spiral component of the cochlea. Now, frequency theory says that your pitch is picked up by how fast your vibrations are hitting your cilia. So it doesn't matter where the cilia is located, just the faster the cilia is moving will dictate the higher the sound or different pitches. Now, you're also going to have the volley principle, which says that pitch that states frequency can cause hair cells to fire in a volley pattern, take turns firing. So the volley means some cells pick up sound, some of them don't, and it just depends on the two. Now, when we talk about hearing, we do have a lot of questions about hearing impairments. So, conduction hearing impairment can result in a damaged eardrum, which prevents your tympanic membrane from vibrating, or damage to the bones in your middle. Now, we also have nerve hearing. So, you have conduction, which means that your eardrum's not working, or your oscilles aren't working, or you have nerve hearing impairment. Can result from damage in the inner ear or damage in the auditory pathways. So anything that is dealing with your cochlea. You can have some surgeries on replacing it. For instance, your cochlear implant is going to be used for your nerve ending impairments, and it's a microphone implant just behind the ear. It picks up sound from surrounding environment. Speech processor selects the ranges in the sound picked by the microphone, and then it converts to a receiver and creates an electrical impulse, which is then processed by your brain, which is pretty cool. And it just attaches to your brain, and it's not common, but it's actually used pretty frequently, which is really cool. Now, taste is your olfactory. Now, taste buds are the taste receptors in your mouth responsible for taste. Keep in mind that taste is your one of your gustation is what we call it here in AP Psych. And there's 5 basic tastes: sweet, sour, salty, bitter, and brothy. Okay? Now, on your tongue, you have different layers and different taste buds. Okay? Now, your sense of smell is olfaction, and olfaction bulbs are located just above the sinus cavity and just below the frontal lobe, and it receives information from the olfactory receptor. Now, there's at least 1,000 olfactory receptors in your brain that are processing your smell. Now, as you can see in this diagram, your olfactory bulb is located very, very, very close to your frontal lobe. Now, your frontal lobe is going to be processing all these smells, which is why when you have a smell, it's going to trigger a very strong emotional response. And this emotional response is going to kind of overwhelm you. So when you smell things, it causes a very strong emotional reaction. So that's why, because it's so close to your frontal lobe. All right, so your somatic senses are your kinesthetic senses. These are your body senses consisting of skin. So your soma, aesthetic, is your feeling of your body. Now, your skin senses are your touch, the pressure, temperature, and pain. All of those types of things are being processed by your somatic senses. And one of the big aspects of your somatic senses is your gate control theory, which is how much pain actually gets to your brain. Sometimes it gets cut off by your brain in order for you to accomplish what you need to accomplish. And it's done by your spinal cord. So your spinal cord is like the gateway into your brain, and it either stops signals from getting in or allows them to get in, depending on the need. Now, when you look at the skin and its receptors, it has pain-sensitive, touch, pressure-sensitive nerves. All of these nerves are embedded into your skin, and they're not everywhere. They're spread out depending on need, especially due to our evolution. Now, sense of touch for different people is strengths are different. For instance, people who are blind, who have lost their sense of sight, typically have a higher somatic sense because they need to be able to distinguish the braille, so they have more sensitive fingertips and stuff like that than people who depend heavily on their eyes. Okay, now we do have some people who have zero sense of pain. They cannot feel anything when they're endangered, and that's a somatic sense at all. Now, another one of your somatic senses is your kinesthetic sense. Now, this is your location of your body in relation to the ground. So what happens is you have different parts of your brain, specifically your vestibular sense. Now, this is your sensation of movement. So some of you are very capable of dancing and twirling and not hitting anybody or walking into something. This is done by your vestibular sense, and it's typically held in your ears, and it deals with information from your eyes. You can get really caught up. Well, I hope you have a wonderful week. I hope you enjoy your senses. Next week it is all theories. So have a great week and good luck.